Hi, everybody. It's Mrs. Mancuso. It is Wednesday, May 13th. So this is our third day of having a different AP question each day. So let's get started. Electrons from the oxidation of glucose and other nutrients by glycolysis in the Krebs cycle are added to the electron-carrying compound NAD plus to form NADH. Ooh, that's what I always call cash money. So you're building up your NADHs. To meet the continuous requirement for NAD plus by cells, NADH is reduced back to NAD plus. Okay, we don't use language like that. You know me, the chemistry person. If you are reducing something back, you're oxidizing it. So it's oxidized through the electron transport chain in the presence of oxygen, that's a plus, or through fermentation in the absence of oxygen. So let me just make another page and let's go back through this again. So in glycolysis, which again happens in all cells. So this is what happens in the cytoplasm. You change glucose into pyruvate, two of them. Essentially, you're breaking it in half. All right. This is a 6-carbon sugar, 1, 2, 3, 4, 5, 6, and this is a 3-carbon sugar. Now, in the process of doing that, it requires 2 ATP to get started, but it makes 4 ATP. So that's a net gain of 2. So if you're a bacteria, you really only have cytoplasm. This is occurring in the cytoplasm, and it's going to give you 2 ATP per glucose. But what also happens is 2 NAD pluses become 2 NADHs. And if my glucose is being oxidized, this is being reduced. My glucose is losing electrons, and those electrons are going from the glucose to NADH. So this is gaining the electrons. All right, and then NADH, which I call my cash money, it now has 2 possibilities of what could happen to it. So you could either do this possibility, which is fermentation. So if there's no oxygen available or no mitochondria available, you have to do anaerobic cellular respiration. So anaerobic means you take this pyruvate and you make it into lactic acid or ethanol based on what enzymes are available, but it does free up the NAD pluses again. So you are oxidizing your NADH, releasing the electrons or losing the electrons and making NAD plus so that at least you could go back, do glycolysis again and make 2 ATP again. Or the other pathway, the other possibility, which is where I definitely call it cash money, this is the aerobic pathway. So the aerobic pathway, you have to have oxygen and you have to have mitochondria. And you're going to change all the pyruvates into CO2, and you're going to use the oxygen and you're going to end up forming water. And what happens here, if I draw a big mitochondria, the NADH is right here. It gets oxidized. Its electrons go through the chain till eventually they make it to oxygen, which will make water. But in that process, it also pumps H pluses into the outer compartment and builds up a nice proton gradient here. And then the proton gradient is used to shuttle the H pluses back through and make a massive amount of ATP. All right, so a little bit of a review of what's going on in terms of NADH, where it's made and what it's used for. All right, back to the question. All right, so here is their little diagram. So their little diagram is showing glycolysis changes NAD plus into NADH, and then fermentation will have no oxygen available, and it will change the NADH back to NAD plus, or the electron transport chain, if you do have oxygen available, will change NADH back to NAD plus. All right, next part of the question. MTND5 is a mitochondrial gene. Okay, mitochondrial gene, another new page. Let's talk about this for a little bit because this is the second time mitochondrial DNA has come up. So we're going to go way back to, here is a prokaryotic cell, and here is the cell that is a precursor to a mitochondria. So a prokaryotic cell took in this other cell, and now this cell is existing inside of another cell. This is our mitochondria, but it is also our first organelle. This process changed this prokaryotic cell into now a eukaryotic cell, and this process is called endosymbiosis. All right, let's talk about the DNA. If I had a prokaryotic cell versus a eukaryotic cell, how is the DNA different? So a prokaryotic cell, first of all, there's no nucleus. You just have one little circular piece. So it is one single circular chromosome. All right, over here, if I have a eukaryotic cell, I have a nucleus and I have pairs of chromosomes. So this is a nucleus with diploid chromosomes. So there are pairs. Over here, there's no pairs. So prokaryotic cells are just haploid. This is just one singular strand of a chromosome. There's no pairs and there's no real nucleus. The difference that is going to be really important in this question is when you think about alleles. Alleles exist on chromosomes. So if I was looking at nuclear DNA, I would have a genotype. I would have two alleles. One came originally from mom, let's say it's a big A. One came originally from dad, let's say it's a little a. That is my genotype for a specific trait. When you have mitochondrial DNA, the mitochondrial DNA, so let's just show another picture here. So here is my eukaryotic cell. Here is my nucleus with pairs of chromosomes. And then here's my mitochondria. It used to be a cell, so it just has circular DNA inside of it. So essentially that DNA is haploid, which means it would only have one letter and it only gets passed on from mom. So let's draw that out. If I have an ovum or an egg here, the egg is going to have a nucleus, the egg is going to have multiple mitochondria, and it only has a haploid nucleus. And then here's my sperm, and inside of here is a nucleus, and it's also haploid. The mitochondria are in what's called the midpiece. So the mitochondria are in here, and when this fertilization happens, the head of the sperm goes in, and that's it. It cuts off. The tail and the midpiece never enter the zygote. So your zygote that forms is going to have now a diploid nucleus, but it still only has mom's mitochondria. And the mitochondria is still only haploid. It's still only single genes. So when you inherit a gene that's mitochondrial DNA, it is only from your mother, which means you can trace it back from every child to their mom, and it is always passed on, and it is always just a single form of a gene. Just one allele, not a pair of alleles. All right, now we can go back to the question. Sorry, I feel like all this analyzing is super worthwhile, so hopefully you're learning a lot as I'm doing this. All right, so this is a mitochondrial gene that encodes a subunit of Nadh dehydrogenase, the enzyme that catalyzes the initial oxidation of NADH to NAD plus and H plus. All right, so I'm going to draw an enzyme. This is my NADH dehydrogenase, and right here would be the spot where NADH fits in. And when it works, it would change this to NAD plus an H plus electrons. That's really the job of oxidation. So NADH dehydrogenase does oxidation of NADH into NAD plus. All right. And it makes sense that that's a mitochondrial gene because it's doing a job that specifically happens in the mitochondria, probably more than any other location. It can obviously happen in the cytoplasm if you have a prokaryotic cell, but this is a really important job to be done in the electron transport chain for aerobic respiration, which happens in a mitochondria. All right, so a mutation in the MTND5 is associated with a rare genetic disorder that results in a buildup of lactic acid. So if you have a certain form of the gene in your mitochondrial DNA, your Nadh dehydrogenase isn't working right and you make more lactic acid. So that means you're not going to be doing as much of the electron transport chain in the mitochondria. Instead, you're probably doing more fermentation. So I'm going to just write fermentation here because every time I see that buildup of lactic acid, you're doing more fermenting. All right, a researcher hypothesized that the mutated Nadh dehydrogenase has decreased activity, but it's not completely non-functioning. Well, that makes sense. If it was completely non-functioning, you wouldn't be able to make so many ATP per glucose and you'd die. So, I mean, it's got to be somewhat functional. If you are surviving, you got to have somewhat function of it. All right, by increasing the pool of NADH in cells, the activity will increase. That's the hypothesis. All right, to test this idea, a researcher treated a group of individuals with the disorder with a vitamin that's similar to NADH and measured the concentration of NAD+ and lactic acid in the blood over the course of 20 weeks. The results are shown in figure 2. All right, let's look at figure 2. So here's figure 2, and it's showing me the concentration of NAD+, and it's showing the concentration of lactic acid. So if I just check this out for a little bit, NAD+ means that NADH is being oxidized and it's making NAD+. So you give them the vitamin on this day and all of a sudden you're making more. So it looks like over maybe 5 to 6 weeks, it really has a really positive effect. But then if they only gave the vitamin one time, after that much time, it appears to have worn off. And then down here, how much lactic acid it's making. So if I do this instead of fermentation, I would expect lactic acid to go down, and look at that. At least in the beginning, when that vitamin is working, it is decreasing the amount of lactic acid made. All right, and it says the concentration of NAD+ in the blood of a representative treated individual. All right, interesting that they're just giving us data from one individual, but all right, we're going to go on with the questions. First question. So first of all, this is a 10-point question. So this is the kind of question that you're going to see as your first question. You will have 25 minutes to answer it and 5 minutes to upload it or submit it, however you want to word it. I suggest always doing these questions right, like typing right in the thing and submitting it just like you've been doing on AP Classroom. And you should be doing the practice ones that are available for you right now. All right, so describe the pattern of inheritance that's most likely associated. So the pattern of inheritance is that this mutant allele is only inherited from the mother. Because it's a mitochondrial gene. All right, so that's how you inherit it. You inherit it from mom because it's only just this mitochondrial DNA. So you only get the mitochondria from mom. So that's how you get it. All right, next question. Explain why individuals are not heterozygous with respect to mitochondrial genes. So they are not heterozygous because it is not a nuclear gene. There are no pairs. So you don't have 2 alleles. You only have 1 allele for a mitochondrial gene because mitochondria mitochondrial DNA is only a single unpaired chromosome. Okay, so because you're getting it from mitochondria, you're just getting one letter that was in that singular DNA. If it was nuclear instead, you'd be getting one from mom, one from dad, and then it could be heterozygous. Heterozygous would be one form of each, but instead you are getting only one allele, whatever allele mom had in her singular strand of DNA in her mitochondria. All right, identify a dependent variable. Okay, so the dependent variable is what is measured in the experiment. What you measure, you always put on the y-axis. So I need a dependent variable. So I go back to the graph, concentration of NAD plus in micromoles per liter. Concentration of NAD plus in U moles per liter. Always make sure that you have a unit. Really, really important. Now there's actually two right answers because there's two different graphs. So you could have also said, or the concentration of the second graph was lactic acid. But I got to check because I think the units were different for the lactic acid. Yes, M moles per liter. All right, so there it is. That's all they asked me to do was identify. So I just have to write what it is. And there are going to be 10 different questions with 10 different task verbs, and it's worth 10 points, which is 65% of your AP score this year. All right, identify one control that the researcher could use to improve the validity of the experiment. All right, all they showed was giving it a vitamin. A control, you want to have that be something that you can use as a comparison. So I want to give a group of people who also have this strange disorder, so also have this rare condition. A placebo. I don't want to give them the vitamin. I want them to think they're taking the vitamin because I want the psychology to be the same, but I want to actually give them a placebo. So give a group of people who also have this rare condition a placebo instead of the vitamin. And also gather their data over the course of the 20 weeks. All right, so that would be a good control to do. That would be something to compare this to. All right, next one. Justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning of and the end of the 20-week period. Okay, so if you gather data over 20 time periods, you're going to have more data. More data to see or notice trends. Over a long stretch of time. Basically, you don't know how long it's going to work for or how long it's going to take to work. And if you think back to the graph went like that. If I only measured it at the beginning and the end, I'd miss all this. So you don't want to miss all that data. So if you measure it many, many times instead, you'll actually see those changes. All right, next question. Describe the relationship. Okay, every time they ask you to describe a relationship, it's always good to put like as this either goes up or goes down, this goes up or goes down. Okay, the relationship between the concentration of NAD plus in the blood. Okay, so let's go back to our graph. What happens to the NAD plus in the blood? Where's our graph? Okay. I'm just going to erase so that I can see more stuff. So NAD plus in the blood, and then I think it actually gave me a time period, first 5 weeks. Okay. So here's my first 5 weeks. So over the first 5 weeks, this increases. And then I go down here and this one, that lactic acid decreases. Okay. So as the NAD plus increases, of course, I would write out the word if I was actually doing the test. The concentration of lactic acid decreases. Now, some of you might think of this also describe the relationship. The relationship is also inversely proportional. You can say one or the other. You don't have to say them both. Proportional. All right, so I like to write it this way because it's showing the relationship. It's showing what happens as one goes up, the other one goes down. All right, next one. Based on figure 2, calculate the average rate of change in blood NAD plus concentration from week 5 to week 17. Okay, if I want a rate, I want change in Y over change in time. Change in time was on the x-axis, so I always do slope. Anytime they ask me for rate, I do slope. So I already know the bottom is going to be from week 17. Whoops, don't know why it's making that black mark there. All right, let's write this again. All right, 17 minus 5. So that's 12. And now I got to go look at how the NAD plus is changing. Okay, so let me get another color here. Week 17 is right here, and I would go across. All right, so let's say that's around 60. And then week 5. All right, so let's say that's 300. So 60 minus 300. So that's negative 240. And you divide and you get negative 20. All right, so first thing, if you do not put that negative on there, it is definitely not going to be right. So negative shows me that the level of NAD plus was going down. It's really, really important that you have that there. All right, and then I also want to make sure I have units. So again, I go back to the graph. So that was U moles per liter per week. These are weeks, right? Weeks, okay. So negative 20. Per week. All right, so that's how I want to make sure I have my answer written, and I want to make sure I definitely have that negative in there because it's showing that it's going down. All right, next question. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of the mutation on the rate of oxygen consumption. All right, so predicting, I just have to say, is it going to go up? Is it going to go down? Is it going to stay the same? What do I think is going to happen? Well, I definitely think that oxygen consumption is going to decrease. So the rate of O2 consumption will decrease. And now the next one is justify it. So now I got to explain what I'm thinking. So I go back to this picture of my mitochondria, and sorry, this is supposed to be a little bit more surface area, but anyways, here was my NADH. And if my NADH does oxidize, it will lose electrons. The electrons will go through the electron transport chain and eventually use oxygen and make water. Well, if my NADH dehydrogenase, if the enzyme that does this work is bad, I am not going to have a lot of this electron transport chain working. If I do not have much electron transport chain working, I don't need as much oxygen. So that's what I have to do here in my justification. So if the NADH dehydrogenase, okay, I'd obviously write out that entire word, but I'm just going to abbreviate it right now, is not oxidizing as much NADH, then fewer electrons will move through the electron transport chain. This will require less O2 as a final electron acceptor. Now, that's really important because you're showing that you understand the use of the oxygen. So the oxygen is used at the end to take the electrons, and if the electrons weren't lost from NADH in the first place, you are not going to need as much oxygen. So that's why I would say it's going down. All right, next one. The researcher had hypothesized that the addition of the vitamin that's similar in structure to NADH would increase the activity of the mutated NADH dehydrogenase enzyme in individuals with the disorder. Explain how the vitamin most likely increase the activity of the enzyme. Okay, so before I start, let's just draw a picture here again. Here is my NADH dehydrogenase, and here is my NADH. It fits right in. But there's not enough of it present, is what it's saying. It's not really being activated. So if my vitamin looks just like this, it will also fit in the space. So because the vitamin also fits in NADH dehydrogenase active site, it will increase the activity of the enzyme. Okay, the way that it works is it makes it seem like there's more substrate available. And that's making it like turn on this enzyme. You just need it to fit in that space. So if the vitamin fits in there, it's going to work. All right. And that was J. So that was our 10th point. All right. So go back through this, see if there's any questions, anything you weren't sure about. I'd be happy to answer your questions. And I hope you guys are doing well. Bye.